Mitosis plays a major role in somatic cells for their division, and by division mitosis is actually helps to grow and healing wounds and plays a many more essential functions in our body. Mitosis actually produces two daughter cells with identical number of chromosomes as their parents. Hello friends, welcome to Biology Concern. I am Animesh. In this video, I am going to discuss about the mitosis in a brief manner. But in my next lecture, I also give you an explanation regarding the molecular basis of kinetochore attachment, chromosome compaction, molecular basis of anaphase movement, and all of the molecular aspects of the mitosis. But in this video, I am stick to the just brief overview with mitosis. So let's jump into the topic. First thing, we have to know about the history of mitosis. So mitosis comes from a Greek word. So it comes from a Greek word, that is the mitos, that means thread. Okay, so it means thread. It comes from a Greek word mitos and it means thread, thread like structure, that's the thread. And the term mitosis coined by German biologist Walther Fleming in 1882. Okay, so Walther Fleming first described about the mitosis. He also described that a thread-like structure that mysteriously appears just before a cell divides into two. So first thing is important about mitogens. Okay, so what are they? Mitogens are specifically some kinds of proteins that are actually essential for a cell to concluding or conducting the mitosis. Mitogens, one of the examples of mitogens is MAPK, that is stands for mitogen associated protein kinase. So MAPK is actually an enzyme. Okay, so it is particularly an enzyme which is a kinase, that means it added phosphate into any kind of structures, any kind of molecule. So in any molecules it actually adds a phosphate group. That is why it is known as kinase. So it is a mitogen associated protein kinase. Those MAPKs are similar or close resembles, so it is close family member of CDKs. Okay, so CDKs is stands for cyclin dependent kinase. Cyclin dependent kinase. So CDK stands for cyclin dependent kinase, and those mitogens or MAPKs, mitogen associated protein kinase, are actually falls under CMGC protein kinase group. Okay, so CMGC protein kinase group is actually a large group of proteins, and it includes MAPKs, CDKs, and also GSK3. Okay, and many more of enzymes in this family, that is the CMGC protein families. MAPK stands for mitogen associated protein kinase, CDK stands for cyclin dependent kinase, GSK3 stands for glycogen synthase kinase 3. Okay, so it stands for glycogen synthase kinase 3. So glycogen synthase kinase, CDKs and MAPKs and many more of the kinases are actually falls under the CMGC kinase protein kinase group. And what are actually main functions of CMGC protein kinase group? They actually adds a phosphate group in any molecules. Okay, so that is the main thing about mitogens and how they activate it. The mitogens are particularly activated by another group of protein that is known as STE protein, and it actually phosphorylates. So it phosphorylates particular residues that is made up of threonine and tyrosine. Okay, so threonine and tyrosine amino acids make a particular loop in the CMGC protein kinase group, and those protein kinase group are actually activated by ST proteins, and ST protein actually phosphorylate those threonine and tyrosine residues, and further they actually activate those mitogens or MAP kinase or CTKs or glycogen synthase kinase 3 or etc. So this is the overview of mitogens and the history of mitosis. Let's jump into the core particle, that is mitosis. Mitosis is further classified into 5 subphases. Okay, so mitosis is classified into 5 subphases. One is prophase, second one is prometaphase, third one is metaphase, fourth one is anaphase, and fifth one is telophase. So what is prophase, prometaphase, metaphase, anaphase, and telophase? What is the characteristic feature? So these 5 stages actually divides the chromosomes. Okay, so divides actually chromosomes, and hence they are known as karyokinesis. So they are known as karyokinesis. Karyokinesis stands for the division of the nucleus. So prophase, prometaphase, metaphase, anaphase, and telophase, those 5 steps are helps the nucleus or central molecule to divide into 2, and after that cytokinesis takes place. And they divide the cytoplasm. So let's jump into the prophase, prometaphase, metaphase, anaphase, and telophase, each of them briefly in this video. So what is the first criteria or first characteristic feature of prophase? Just consider this cell. In first step, prophase, the nuclear membrane dissolution takes place. That means the nuclear membrane disappearing in this position or in this phase of prophase, but it is the beginning to dissolve the nuclear membrane. Okay, chromatin, and in this phase now compacted in the form of chromosomes. So chromatin compacted in this phase in the form of chromosomes. Nucleolus already disappeared, along with Golgi bodies, mitochondria, and other cellular organs are also disappeared in this stage, and duplicated centrosome, that is consisted of the centrioles, they are moving apart from each other and they are moving towards the pole. So that is the characteristics features of prophase. In this phase, first thing is chromosome compaction occur. Okay, so chromosome compaction, that is the condensation of the chromatin fiber. In my next video, I will cover about the chromosome compaction in details. Second thing is nuclear envelope, nuclear membrane dissolution takes place. Okay, so takes place. Chromosome compaction occurs, nuclear membrane dissolution takes place, and third one, centrosomes move. Centrosome moves towards the pole. That is the third point of prophase. This is the characteristic feature of particular prophase. So now we jump into the prometaphase. What is the prometaphase stands for? So in this case, nuclear dissolution is complete. Nuclear envelope is completely disappeared. So the major difference between prophase and prometaphase. So this one is the prometaphase, and some of the books refer it as a late prophase. So first criteria of this prometaphase is the complete dissolution of nuclear envelope. So complete dissolution of nuclear membrane. So nuclear membrane is completely dissolved and centrosomes are beside each of the opposite poles of the cells. So here centrosomes are resides opposite poles of the cell. Okay, so that is the another characteristic feature. And chromosomes are occurred present in the cell but in a unorganized matter. So here chromosomes appears in the cell but they are unorganized. And third thing is kinetochore assembly takes place. So region kinetochore is a special region. So kinetochore is a special region that actually produces in the centromere region of chromosome, and it's actually made up of more than 100 proteins. Okay, so this region is known as kinetochore region. So this is known as a kinetochore region. It actually helps the spindle fiber to bind with particular pair of chromosomes. Okay, so this particular region is stands for kinetochore region. Okay, so this region is known as kinetochore region. And in this case, in the prometaphase, the spindle fibers comes out from the centrosome and they binds with the particular kinetochore by a process known as search and capture model. That means those spindle fibers are actually originated from the centrosomes and they move towards the cell. Okay, towards the cell, and it actually search for particular kinetochore region. When it actually find a kinetochore, then it binds with the kinetochore and it makes them with the centrosomes together. Okay, so here spindle fibers are actually finds the kinetochore and they locks the microtubule with the kinetochore. That means it actually locks the centrosome with the chromosome by the help of those spindle fibers. But all of the spindle fibers cannot attach with the particular kinetochore region because there are 46. In case of mitosis, there is 46 pair of chromosome presence. Okay, so in this case we have only 46 pair of kinetochores. So 46 kinetochores are adjusted with 46 spindle fibers. But in case of this cell, in this prometaphase, many more spindle fibers are making up, and they extends from one pole to another. So it is not possible for all spindle fibers to bind with the particular 46 pair of kinetochores. Okay, our next point is metaphase. Okay, so in this case metaphase actually occurred in a very tiny amount of time. And what actually metaphase done in metaphase, the chromosomes are organized in a central equatorial plane. Okay, so all of the chromosomes are in equatorial plane in this case, and those equatorial plane are known as metaphase plate. So arranging of chromosomes arranging of chromosomes in the metaphase plate, that is the main event, takes place during the metaphase. Okay, so here you observe that in prometaphase chromosomes are organized here and there in the cell, but in case here chromosomes are organized in a particular planar or equatorial region of this particular cell. Okay, so what is the difference between this and this? In this case, prometaphase, in an early metaphase, those spindle fibers create a force, that is a pulling and pushing force, by which they actually shifted those particular pair of chromosomes in the middle of the cell, and those middle portion of the cell known as metaphase plate. So in metaphase, it actually organizes the chromosomes in a particular manner. Okay, so that is the function of metaphase. Now our fourth phase is anaphase. So in this case, actually chromosomes are separated from each other. That is, the chromosomes are separated from each other with the help of some of the proteins or some of the enzymes. The sister chromatids present in the metaphase plate are actually held together by a protein known as securin. Okay, so securin is the main protein, or securin is the main enzyme that is actually helps to hold together those sister chromatids together, and it actually plays a crucial role for cohesin proteins or cohesin molecules. So those cohesin molecules are actually particularly bind those two sister chromatids together. But in case of anaphase, there is another enzyme known as separase. So separase secretes. Okay, in this case, separase now synthesize and secretes inside of the cell, and those separates are bind with the securins. So if that is the separase, so it binds with particular securin molecules or securin proteins, and it actually inhibit the function of securins. Okay, and now it cleaves the cohesin molecules or cohesin proteins from the joining of the two sister chromatids, and after that the two centrosomes create a pulling force, and now the chromosomes are being separated in a orderly manner. Okay, so in this case, particular pair of chromosome now separated, and single chromosome is make a make a movement towards the particular pole. So this is the anaphasic movement of the particular stage. So this is the anaphasic movement of particular chromosome or particular pair of chromosome. With the help of separase, it actually binds with securin and inhibits the function of cohesin that holds two sister chromatids together. And when there is no securin, cohesins are cleaved off from the sister chromatids, and now chromosomes are being separated towards the pole. That is the movement, that is known as anaphasic movement. Okay, so in next phase, what happens in the telophase? In the telophase, so in case of telophase, nuclear membrane appears here, and chromosomes are already separated here. And there is a cleavage furrow produces in this telophase portion. Okay, so in telophase, inbound movement of the cell membrane or plasma membrane occurred, and it appeared like a cleavage furrow, and it did produce a cleavage furrow, and in here nuclear membrane again reappears along with mitochondria, Golgi bodies, and centrosomes are present already in there. So cleavage furrow produces, nuclear membrane reappeared, nucleolus already also reappeared, and chromosomes are now segregated during this phase. This is the completion of mitosis. Okay, now cell enters into the cytokinesis. So in cytokinesis, what happens? Those cleavage furrow that actually produces in the telophase actually move towards further, and they progress towards each other, and ultimately at a point they actually joins with each other and produces two cell that contains the chromosomal content identical with their parent cell. So this is the brief idea about mitosis, what mitosis is, what the mitogens are, and prophase, prometaphase, metaphase, anaphase, telophase, and what happened in the case of cytokinesis. So I think this video is really helpful for you to understand the total process in a brief manner. If you like that video, please hit the like button. Don't forget to subscribe, and thanks for watching.